Welcome to Key to My Research, the science podcast by the Excellence Strategy at the University of Tübingen. In this podcast, we explain in a simple way how outstanding scientists are researching complex topics that affect our everyday lives. My name is Jennifer and I'm your host. In today's episode, we meet Professor Juliane Walz.
Very, very early in my life, I decided I wanted to do something with cancer and develop treatments for cancer. This started when my mom suffered from breast cancer when I was 15 years old.
She's Professor for Peptide-based Immunotherapy and Medical Director of the Clinical Cooperation Unit, Translational Immunology. Her research focuses on the development of novel peptide-based immunotherapeutic concepts for tumor and infectious diseases.
I see this high need every day as a clinician in the hospital. Of course, I have this personalized experience with cancer, and I think this drives me every day to performing better and providing novel options, because it's really hard to enter a patient room and tell the patient we have nothing more to give. And I really hope that there will be a time point where I don't have to say this again to a cancer patient.
As we know, this is not yet possible, but to achieve this, Professor Walz is mainly developing vaccines for cancer patients. These are based on peptides. Peptides are short proteins found on the surface of all cells in the human body, including cancer cells.
And these peptides differ between cancer cells and healthy cells and allow the immune system to detect tumor cells as foreign and to destroy them. So my research really focused on identifying these peptides and using them for immunotherapy to target cancer cells.
To understand why Professor Walz's research is so important, let's take a closer look at immunotherapy.
Immunotherapy absolutely has entered now clinical routine and has really added a layer of therapy to the classical cancer treatment, which is surgery, radiation, and chemotherapy. And this was started with the development of the so-called immune checkpoint inhibitors, which I think was claimed as the breakthrough in cancer research already in 2013. So immune checkpoint inhibitors indeed are antibodies that are able to unlock the potential of the immune system, and namely the T cells, which are the strongest cells of our immune system to attack cancer. And these immune checkpoint inhibitors just unlock the activity of these T cells to target cancer cells. However, they do it in an unspecific way. And based on this success of the immune checkpoint inhibitors that are now approved for the treatment of several cancers, the specific cancer immunotherapies that really target the immune cells directly to the cancer emerged, which are the bispecific antibodies, which are the CAR T cells, and which are now the cancer vaccines that are now absolutely in the focus of research. What cancer vaccines do, and you always have to see it in contrast to the prophylactic vaccines that we are applying against pathogens, against virus and bacteria, is that they are applied to patients that are already suffering from cancer. And the idea is with the vaccine to train the immune system, and particularly the T cells, to detect the antigens that are presented by a tumor and then attack the tumors. So usually you go at a site far away from the tumor where you have no tumor cells and no immunosuppressive microenvironment that is usually around the tumor to train the immune system. You apply antigens or mRNA or genetic parts of the tumor cells, and you train the immune system locally.
Peptide vaccines represent a huge opportunity for scientists and patients. The vaccines can now be developed individually for each patient, something we call personalized medicine. So it fits the patient's body exactly and has a high chance of producing the desired results with minimal side effects.
We really have the opportunity to analyze individual patients' tumor and characterize these peptides from really the individual tumor cells of the patient, and then use these peptides in an individualized approach for a vaccine. So this really means we analyze the peptides, we produce them individualized for the patient, and then apply a personalized vaccine. And there are, of course, various steps to personalize. Of course, we could also only personalize to the specific HLA allotype, which is comparable to the blood group of a patient. So it's an individual marker of the patient. And we could just select peptides for these HLA allotypes. That would be the smallest level of personalization. What we also could do, and this is what we also do in a trial, is a so-called warehouse concept, where we produce the best peptides that we have analyzed before in drug quality, and then do these individualized analysis of the patient's peptide, and then select out of this pre-produced warehouse the best peptides and compose them in a personalized vaccine, which makes it much faster and cost-saving than really producing each individual peptide for each individual patient.
In contrast to chemotherapy, in which large areas of diseased and healthy cells are destroyed and the entire organism suffers, individualized cancer vaccines only target the cancer cells using the patient's own immune system. But the cancer vaccine takes longer to show results.
The vaccine first has to train the T cells, which takes about 4 weeks, I would say. Then T cells have to expand, and then they have to find the tumor cells. So until you see effects, I would say it takes 8 weeks to 3 months.
There is one current project that Professor Walz and her team are working on. It is a vaccine against a cancer called fibrolamellar carcinoma. This is a rare disease that usually affects the liver of young adults or adolescents with no previous liver disease.
In most cases, this disease is detected late. So this means when the cancer has already spread and there are metastases. So the cancer is not treatable by surgery. And indeed, so far, there is no approved treatment for this specific tumor entity. And what we've done already in preclinical work is we have developed a vaccine that is targeting a specific genetic alteration in these cancer patients. And we are currently evaluating this specific vaccine in a clinical trial. And I really hope that this will change the treatment landscape of this rare cancer where there's nothing available for these really young adults. We have one case where we applied already this vaccine to a 15-year-old girl that was in a situation where there was no treatment left and she suffered from this fibrolamellar cancer. And we were asked if there's any possibility what we could do. And we designed this vaccine for that patient and applied only 2 doses and could show that we induced a strong T-cell responses against the vaccine peptides. And since we applied this vaccine and induced the T-cell responses, the patient had no relapse anymore. So she's disease-free, which is now 3.5 years since the last dose of the vaccine. And this is something what really drives all of us and enabled also then this clinical trial where we could now offer this vaccine to many more of these patients. And I really hope that this example in a rare tumor entity could be spread to other solid tumors, breast cancer, lung cancer, to make these vaccines just available for more patient groups. I think the idea of combining anti-cancer drugs to improve immune system activation and clinical efficacy, that is really one of my goals that I want to follow up in the next years. And I think Tübingen provides the perfect environment to reach these goals. And there are so many preclinical work going on. And we hopefully will next year also enter the first trial where we combine for the first time the bispecific antibodies and peptide vaccines. I think we are really far also in providing these approaches to the cancer patients now.
At the University of Tübingen, Professor Walz is one of the 3 spokespersons of the Image-Guided and Functionally Instructed Tumor Therapies, IFIT, Cluster of Excellence. It is the only oncology cluster in Germany with 3 research areas working together on an interdisciplinary basis. We already talked about this cluster of excellence in the episode with neuro-oncologist Professor Tabata Bay. You can listen to the episode in our podcast channel. For Professor Walz, the cluster is a perfect way to develop and advance her research.
Tübingen really builds on this pioneer work and has established really a strong immunology and cancer research community, which was further strengthened by our cluster of excellent IFIT that was started in 2019. And it really merged the 3 main research areas that we have in Tübingen. So imaging, making a tumor cell or tumors visible for the physicians, and molecular target therapy, so which is the direct specific targeting of tumors by drugs, and immunotherapy, which is really harnessing the immune system against cancer. And I think this really strong community that is now merged in this cluster really helps us to develop novel treatments and then also directly translate them to the patients. The University of Tübingen really provides me the perfect background to do my science. And also starting as a young physician in clinics and wanting to start a scientific career, there are so many great opportunities to support young researchers. I entered the Fortune program, that it's an internal program that supports first staff and funding to start research projects. There are also a lot of programs for females in science. So I was really supported in the so-called TÜV program, supporting females on an academic track to habilitation and getting a professorship. Also with regard to funding, but also mentoring, learning all these skills and knowledge that you need to do an academic career. There, the University of Tübingen really provides the perfect environment.
This was Key to My Research, a podcast produced by Changing Time in cooperation with the University of Tübingen. For more information and the links to the sources, see the show notes below. There, you can also find a link to the whole interview with Professor Walz. If you enjoyed this episode, make sure to subscribe to the podcast, leave a review, and recommend it to your friends and colleagues. Authors Chris Veit, Jotti Fotiades. Special thanks to Professor Juliane Walz, Heiko Heil, Oliver Häusler, Kurt Schneider, and Oliver Lichtwald. My name is Jennifer. Thanks for listening, and see you next time.